Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food or they need, and all other organisms depend on them for their food, for their needs. The green plants make or rather synthesize the food they need through photosynthesis, and they are called they are therefore called autotrophs. You have already learned that autotrophic nutrition is found only in plants, and all other organisms depend on the green plants for their food are heterotrophs. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is responsible for the release of oxygen into the atmosphere by green plants. How have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthesis, photosynthetic machinery, and the various type of reaction that transforms light energy into chemical energy. 13.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in earlier classes have you shown that chlorophyll in bracket green pigment of the leaf bracket complete light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf and a leaf that was partially covered with black pepper and exposed to sunlight. On testing these leaves on the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaf in the presence of light. Another experiment you may have carried out where a part of a leaf is enclosed in a test tube containing some KOH soak cotton which absorbs CO2, while other half is exposed to air. The setup is then placed in light for some time. On the testing for the presence of starch later into plants of the leaf, you must have found that the exposed part of the leaf tested positive for starch while the portion that was in the tube tested negative. This shows that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? 13.2 Early experiment. It is interesting to learn about these simple experiments that lead to a gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770, performed a series of experiments that reveal the essential role of air in the growth of green plants. Priestley, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesis as follows: plant restore the air whatever the breathing of animals and burning of candles remove. Can you imagine how Priestley would have conducted the experiments using a candle and a plant? Remember, he would need to rekindle the candle or to test whether it burns after a few days. How many different ways can you think of it light candle without disturbing the setup? Using a similar setup, one used by Priestley, but placing it once in the dark and once in the sunlight, Jan Ingenhouse in 1730 to 1799 showed that sunlight is essential for the plant process that somehow purifies the air fouled by burning candles or breathing animals. Engine house and elegant experiment with an aquatic plant show that in a bright sunlight, a small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when the plant grew. Glucose is usually stored as a starch. His later studies show that the green substance in a plant's chlorophyll as we know it now. It is located in a special body, later called chloroplast. Within the plant cell, he found that the green parts of plant is where glucose is made, and that glucose is usually stored as a starch. Now consider the interesting experiment done by T.W. Engelmann in 1843 to 1909. Using a prism, he split light into its spectral components that and then illuminated a green alga. Cladophora, a place in a suspension of aerobic bacteria. The bacteria were used to detect the site of O2 evolution. He observed that the bacteria accumulate mainly in the region of blue and red. He split the spectrum. The first action spectrum of photosynthesis was thus described. It resembles roughly the absorption of spectra of chlorophyll A and B, discussed in section 13.4. By the middle of 19th century, the key features of a plant of photosynthesis were known, namely the plants could be used light energy to make carbohydrate from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as CO2 plus H2O gives CH2O plus O2 in presence of light, where CH2O represented as carbohydrate, example glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist, Cornelis van Niel, 1897 to 1985, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 in presence of light gives 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis. When H2O is instead of the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur and sulfate depending on the organisms and not O2. Hence, he inferred that O2 evolved by green plants comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic technique. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O in presence of light gives C6H12O6 plus 6CO2 plus 6O2, where C6H12O6 represents as glucose. The O2 release is from water. This was proved using radioisotopic technique. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as a substrate are used in the equation given? 13.3. Where does the photosynthesis take place? You would of course answer in the green leaf or in the chloroplast based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does takes place in green leaves of the plants, but it does so also in green parts of the plant. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along with the walls of mesophyll cells such that they get optimum quantity of the incident light. When do you think the chloroplast will align with their flat surfaces parallel to the walls? Then what then would they are perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stromal lamellae, and the matrix stroma. There is a clear division of labor within the chloroplast. The membranous system is responsible for trapping of light energy and also for the synthesis of ATP and NADPH. In stroma, the enzymatic reaction synthesizes sugar, which in turns from starch. The former set of reactions, since they are directly light driven, are called as light reaction, photochemical reactions. The latter are not directly light driven but are dependent on the products of light reaction ATP and NADPH. Hence, to distinguish the latter, they are called as, by convention as dark reaction. Carbon reactions, however, this should not be constructed to mean that they occur in darkness or that they are not light dependent. 13.2 Diagrammatic representation of an electron micrograph of a section of chloroplast. 13.4 How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green and their leaves? Even in the same plant, we can look for answer to this question by trying to separate the leaf pigment of an green plant through a paper chromatography. A chromatographic separation of a leaf pigment shows that the color that we see in leaves is not due to a single pigment but due to four pigments: chlorophyll A, bright or blue green in the chromatogram, chlorophyll B, yellow green, xanthophylls yellow, and carotenoids yellow to yellow orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light and specific wavelength. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A and chlorophyll A pigment to absorb light of different wavelengths. Figure 13.3a. Of course, you can familiar with the wavelength of a visible spectrum of light as well as visual. From the figure 13.3a, can you imagine you determine the wavelength color of light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelength too? If yes, which one? Now look at the figure 13.3b showing the wavelength at which the maximum photosynthesis occur in a plant. Can you see that the wavelengths at which there is a maximum absorption by chlorophyll A, that is in the blue and the red region, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of a chlorophyll A and the action spectrum of photosynthesis? These graphs together show that the that the most of the photosynthesis takes place in the blue and the red region of the spectrum. Some photosynthesis does takes place at the other wavelengths of the visible spectrum. Let us see how this happens. Through chlorophyll is a major pigment responsible for trapping of light. Other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called accessory pigment, also absorb light and transfer the energy to the chlorophyll A. Indeed, they are not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect the chlorophyll A from photooxidation. 13.5. What is light reaction? Light reaction or the photochemical phase include light absorption. What is water splitting, oxygen release and the formation of high energy chemical intermediates. ATP and NADPH, several proton complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complex LHC within the photosystem first PS first and photosystem second PS second. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigments of molecule bound to photoproteins. Each photosystem has all pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. Figure 13.4. These pigments helps to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystem. In PS first, in the reaction center, chlorophyll A has absorption peak at 700 nanometer. Hence, it is also called P700. While in PS second, it has absorption maxima at P680 nanometer and is also called as P680. 13.6. The electron transport system. In photosystem second, the reaction center of chlorophyll A absorbs 680 nanometer wavelength of red light, causing electron to become excited and jump into an orbit further from the atomic nucleus. These electrons are picked up by an electron acceptor, which is passed to them to an electron transport system containing cytochromes. Figure 13.5. This movement of electron is downfield in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but they are passed on the pigments of photosystem first, PS first. Simultaneously, the electrons in the reaction center of PS first are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then move downhill again, this time to a molecule of a green energy rich NADP plus. The addition of these electrons reduces NADP plus to NADPH plus H plus. This whole scheme of transfer of electrons starting from the PS second uphill to the acceptor down the electron transport chain to PS first, excitation of electrons, transfer to another acceptor, and finally downhill to the NADP plus, reducing it to NADP plus H plus is called Z scheme. Due to its characteristic shape, figure 13.5, this shape is formed when all the carriers are placed in a sequence on a redox potential scale. 13.6.1 Splitting of water. You would then ask, How does PS second supply electron continuously? The electrons that were moved from the photosystem second must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with PS second. Water is split into 2H plus O and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from the photosystem first and provided by photosystem second. 2H2O plus gives 4H plus O2 plus 4 electron. We need to emphasize here that the water splitting complex is associated with PS second, which is itself is a physically located on the inner side of the membrane of the thylakoid. Then where are the protons and O2 formed, likely to be released in the lumen or on the outer side of the membrane. 13.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability to extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in the chemical bond. The process through which the ATP is synthesized by cells in mitochondria and the chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from EDP and inorganic phosphate in the presence of light. When two photosystems work in a series, first PS second and then PS first, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain. As seen earlier in the Z scheme, both ATP and NADP plus H plus synthesized by this kind of electron flow. Figure 13.5. When PS first is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could happening in the stroma lamellae, while in the membrane or lamellae of the gramina have both PS first and PS second. Stroma lamellae membranous lack PS second as well as NADP reductase enzyme. The excited electron does not pass to an NADP plus but is cycled back to the PS first complex through an electron transport chain. Figure 13.6. The cyclic flow hence results only in the synthesis of ATP but not NADP H plus. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for the excitation. 13.6.3 Chemiosmotic hypothesis. Let us now try to understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism likely in the respiration or in the photosynthesis too. ATP is synthesized and linked to development of a proton gradient across the membrane. These are the membranes of thylakoids. There is one difference through here the protons accumulate is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria. When electrons move through ETS in chapter 14, let us try to understand what are the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electron and their transport to determine the steps that cause a proton gradient to develop. Figure 13.7a. Since splitting of water molecule takes place in the inner side of the membrane, the proton and or hydrogen ions that are produced by the splitting of water accumulate within the lumen of a thylakoid. B. Electron move through photosystem. Protons are transported across the membrane. This happens because the primary acceptor electron, which is located towards outer side of the membrane, transfer its electron not to an electron carrier but to an H plus carrier. Hence, this molecule remove a proton from the stroma while transporting to an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane along with the electrons that come from the acceptor of electron of PS first. Protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are removed from the stroma. Hence, within the chloroplast, the protons in the stroma decreases in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane, as well as a measurable decrease in the pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane into the stroma through the transmembrane channel to the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of a proton across the membrane. The outer portion is called as CF1 and the protrudes on the outer surface of the thylakoid membrane on the side of that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle and the ATP synthase, which makes the enzyme synthase several molecules of energy pack ATP. Chemiosmosis requires a membrane, a proton pump, and proton gradient. ATP synthase energy is used to pump a proton across a membrane to create a gradient and a high concentration of proton within the thylakoid lumen. ATP synthase has a channel that allows diffusion of protons back across the membrane. This release through energy to activate ATP synthase enzyme that catalyzes the formation of ATP along with NADPH produced by the movement of electron. The ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing of CO2 and synthesis of sugars. 13.7 Where are the ATP and NADP used? We learned that the products of light reaction are ATP, NADPH and O2. If of these, O2 diffuses out the chloroplast, while ATP and NADP is required to drive the process leading to the synthesis of food, more accurately sugars. This is biosynthetic phase of photosynthesis. This process does not directly depend on the presence of sunlight, but it is dependent on the products of light reaction, that is ATP and NADPH. Besides CO2 and H2, you may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic phase continues for some time and then stops. If light is made available, the synthesis starts again. Hence we say that calling the biosynthetic phase as the dark reaction is a misnomer. Discuss this amongst yourselves. Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O bracket N or sugars. It was interest to scientists to find out how this reaction proceeds, or rather, what was the first product formed when CO2 is taken into the reaction or fixed. Just after the World War II, among the several efforts to put radioisotope to beneficial use, the work of Melvin Kelvin is exemplary. The use of radioactive 14C by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He was contributed to working out complete biosynthetic pathway, hence it was called as Kelvin cycle. After him, the first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? It have 3 carbon atoms. Scientists also tried to know whether the all plants have PGA as the first product of CO2 fixation, or whether any other product was formed in the other plants. Experiment conducted over a wide range of plants led to the discovery of another group of plants, whether the first stable product of CO2 fixation was again an organic compound, but one which had 4 carbon atoms in it. This acid was instead identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: those plants in which the first product of CO2 fixation is C3 acid, PGA, that is C3 pathway, and those in which the first product was as a C4 acid, or OAA, that is C4 pathway. And these two groups of plants show that other associated characters that we will discuss later. 13.7.1 The primary acceptor CO2. Let us now ask ourselves a question that is asked in the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule which, after accepting or fixing CO2, would have 3 carbons of PGA? The studies unexpectedly show that the acceptor molecule was a 5-carbon ketose sugar, ribulose biphosphate, RUBP. Did any of you think this is possibility? Do not worry. The scientists also took a long time to conduct many experiments to reach this conclusion. They also believed that since the first product was C3 acid, the primary acceptor would be 2-carbon compound. They spent many years trying to identify a 2-carbon compound before they discovered a 5-carbon RUBP. 13.7.2 The Kelvin cycle. Kelvin and his co-workers then work out the whole pathway and show that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Kelvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that Kelvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathway. For ease of understanding, the Kelvin cycle can be described under 3 stages: carboxylation, reduction, and regeneration. First, carboxylation. Carboxylation is the fixation of CO2 into stable organic intermediate. Carboxylation is the most crucial step of the Kelvin cycle, where CO2 is utilized for the carboxylation of RUBP. The reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP, carboxylase oxygenase, or Rubisco. Second step, reduction. These are These are the series of reactions that led to the formation of glucose. The step involved utilization of two molecules of ATP for the phosphorylation and two of NADPH for reduction per CO2 molecule. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of one glucose molecule from the pathway. Third, regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration step requires one ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering in the Kelvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is possible to meet these differences in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make one molecule of glucose, 6 turns of the cycles are required. Work out how many ATP and NADPH molecules will be required to make one molecule of glucose through the Kelvin pathway. It might help you understand that all of these, if we look at what goes in and what comes out of the Kelvin cycle in 6 CO2, 18 ATP and 12 NADPH, and out 1 glucose, 18 ADP and 12 NADP. 13.8 The C4 pathway. Plants that are adapted to dry tropical regions have C4 pathway mentioned earlier. Though these plants have C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Kelvin cycle as the main biosynthetic pathway. Then, in what way are they different from C3 plants? And this is a question that you may reasonably ask. C4 plants are special. They have special type of leaf anatomy. They tolerate higher temperatures. They show a response to a high light intensities. They lack a process called photorespiration and have a greater productivity of biomass. Let us understand these one by one. Study of various sections of leaves of one of C3 plant and other is a C4 plant. Do you notice the differences? Do both have same type of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 plants are called bundle sheath cells, and the leaves in which have such anatomy and are said to have a Kranz anatomy. Kranz means wreath and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles, and they are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of leaves of C4 plants, maize or sorghum, to observe the Kranz anatomy and the distribution of mesophyll cells. It would be interesting for you to recollect the leaves and diverse species plants around you and cut vertical sections of the leaves. Observe under the microscope and look up for bundle sheath cells around the vascular bundle. The presence of bundle sheath would help you identify the C4 plants. Now study the pathway shown in the figure 13.9. This pathway that has been named Hatch and Slack pathway is again a cyclic process. Let us study the pathway by listing steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate PEP and present in the mesophyll cells. The enzyme responsible for the fixation of a PEP carboxylase or pepcase. It is important to register that mesophyll cells lack Rubisco enzyme. The C4 acid is oxaloacetic acid is formed in the mesophyll cell. It then forms other 4-carbon compound like malic acid or aspartic acid in the mesophyll cell itself, which are transported to the bundle sheath cells. In bundle sheath cells, these C4 acids are broken down to release CO2 and 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll cell, where it converted into PEP again. Thus completing the cycle. The CO2 released in the bundle sheath cells enters the C3 or the Kelvin pathway, a pathway common for all plants. The bundle sheath cells are rich in enzyme of ribosco ribulose biphosphate carboxylase oxygenase ribosco, but lack in peptidase. Thus is the basic pathway and that results in the formation of sugar. The Kelvin pathway is common to the C3 and C4 plants. Did you note that the Kelvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cell, but does so only in the bundle sheath cells. 13.9 Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants: photorespiration. To understand photorespiration, we have to know a little bit more about the first step of Kelvin pathway, the first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form two molecules of 3-PGA that is catalyzed by ribosco. RUBP plus CO2 in presence of ribosco gives 2 into 3-PGA. Ribosco, that is the most abundant enzyme in the world. Do you wonder why? It is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? Ribosco has a much greater affinity for CO2 when the CO2 O2 is nearly equal for O2. Imagine what would happen if there were not so. This binding is competitive. It is the relative concentration of O2 and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some O2 binds to ribosco, hence CO2 fixation is decreased. Here the ribosco, instead of being converted, two molecules of a PGA binds with O2 to form one molecule of phosphoglycerate and phosphoglycate phosphoglycode 2 carbon in pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugar nor the ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP and NADPH. The biological function of photorespiration is not known yet. In C4 plant, photorespiration does not occur. This is because they have a mechanism that increase the concentration of CO2 at the enzyme site. This takes place when C4 acid from mesophyll cell is broken down in the bundle sheath cells to release CO2. This results in increasing the intracellular concentration of CO2 in turn. This ensures that the ribosco function as a carboxylase is minimizing the oxygenase activity. Now that you know that the C4 plant lack photorespiration, you probably can understood why productivity and yields are better in these plants. In addition, these plants show tolerance to a higher temperature. Based on the above discussion, can you compare the plants showing the C3 and C4 pathway? Use the table format given and fill in the information. 13.10 Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plant. Photosynthesis is under the influence of several factors, both internal plant and external. The plant factors include the number, size, age, orientation of leaves, mesophyll cells, and the chloroplast. Internal CO2 concentration and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of plant. The external factors would include the availability of sunlight and temperature and CO2 concentration and water as a plant photosynthesis. All these factors will simultaneously affect its rates. Hence, though several factors interact and simultaneously affect photosynthesis or CO2 fixation, usually one factor is a major cause or is one that limits the rate. Hence, at any point, the rate will determined by the factor available at suboptimal level. When several factors affect any biochemical process, Blackman's in 1905 law of limiting factor comes into effect. This states following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantities change. For example, despite the presence of a green leaf and the optimal light and CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf, if given optimal temperature, will start photosynthesizing. 13.10.1 Light. We need to distinguish between the light quantity, light intensity, and duration of exposure to the light. We'll discussing light as a factor that affects the photosynthesis. There is a linear relationship between incident light and CO2 fixation rate at the low intensities. At higher light intensities, gradually the rate does not show further increase, or as other factors become limiting. What is the interesting to note that light saturation occurs at 10% of the full sunlight. Hence except for the plants in shades or in dense forest, light is rarely a limiting factor in a nature. Increases in incident light beyond a point cause breakdown of chlorophyll and a decrease in photosynthesis. 13.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in the concentration up to 0.5% can cause an increase in CO2 fixation rate. Beyond these levels can become damaging over longer period. The C3 and C4 plants are differently to CO2 concentration. At low light conditions, neither a group responsible to high CO2 conditions. At high light intensities, both C3 and C4 plants show increase in the rate of photosynthesis. What is important to note that is the C4 plant show saturation at about 360 ml, while C3 is responsible to increase CO2 concentration and saturation is seen only beyond 450 ml. Thus, current availability of CO2 levels is limiting to the C3 plant. The fact that C3 plants respond to a high concentration by showing increased rate of photosynthesis leading to a high productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. 13.10.3 temperature. The dark reaction being enzymatic are temperature controlled. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. The C4 plant respond to a higher temperature and show a higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to a temperature climate. 13.10.4 Water. Even though water is one of the reactants in the light reaction, that the effect of water as an factor is more through its effect on the plant rather than its directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by leaves through stomata and used for making carbohydrate. Principally glucose and starch. Photosynthesis takes place only in green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are site for light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages: the light reaction and the carbon fixing reaction. In the light reaction, the light energy is absorbed by the pigment present in the antennae. The funnel to a special chlorophyll A molecule called reaction center chlorophyll. There are two photosystems, PS first and PS second. PS first has a 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS second has a P680 reaction center that absorbs red light at 680 nanometer. After absorbing light, electrons are excited and transferred through PS second and PS first, and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to movement through the F0 part of the ATP enzyme release enough energy for synthesis of ATP. Splitting of water molecule is associated with PS second, resulting in the release of O2. Protons and transfer of electrons to PS second. In the carbon fixation the cycle in the carbon fixation cycle, CO2 is added to the enzyme Rubisco to a 5 carbon compound RUVP that is converted to 2 molecules of 3 carbon PGA. This is then converted by sugar by the Kelvin cycle, and the RUVP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful and oxygenation reaction in the C3 plant. Photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4 carbon compound. In the bundle sheath cells, the Kelvin pathway is carried out for the synthesis of carbohydrate. Thanks. Subscribe to NEET audio channel for